Hello. I am sorry to disappoint everyone who was hoping for a really extensive design tree and transgenerational plasticity, but I thought I'd change it up this year and not talk about that. This is a project that I've been working on for the past few years in collaboration with Rebecca Fox at UTS and now at ANU, and I'll thank her again at the end. So we all know that the environment is changing and that we're concerned about the environmental change that's going to occur globally and on coral reefs. And we're concerned about the fact that the average temperature is going to change, and also that thermal anomalies are potentially going to become more frequent events. So it's not just the average change, but also these extreme temperature conditions that we're concerned about. And why we're concerned about it for marine organisms in general, but especially coral reef fish, is because they're ectothermic and they therefore lack internal regulation, which means that their cellular function, performance and fitness is tightly linked to environmental temperature. And what's becoming more of a common trend in lizards, in coral reef fish, and a lot of species, ectothermic species, is that tropical species tend to have a much narrower thermal performance curve, and we're finding generally that they're living a lot closer to their thermal maximum, which is definitely a concern as we're seeing average increases as well as increases in thermal anomalies. So in terms of what we know for coral reef fish, a lot of it is in terms of laboratory research. So we go out to the reef, we collect different species, we bring them back to the lab, and we put them under future projections or just extreme temperatures to see how they respond. And generally what we're finding is that for most coral reef fish, they're living somewhere near their thermal optimum during summer. When we put them under increases in thermal stress, we see that performance or fitness declines. But what we really wanted to do with this project is take it from the laboratory out into the field and start asking questions about species in their natural environment. And firstly, we wanted to understand what are the range of body temperatures that coral reef fish experience in the field, and is there any evidence that a coral reef fish can behaviorally thermoregulate? And so the expectation if you have no thermoregulation is that you would experience the full seasonal temperature range. And over years and generations, then you may get a situation where your thermal performance curve is adapted to that full range of conditions. Your optimum is somewhere in the middle or somewhere near where you mostly experience seasonally. Thermoregulation allows a species to narrow the range of thermal conditions that it experiences, and over evolution has the potential to then narrow the thermal performance curve and potentially shift that optimum to somewhere different than the full seasonal range. They then therefore may not experience body temperatures at the top or the bottom of the seasonal range. And a lot of what we know about behavioural thermoregulation comes from lizards and terrestrial ectotherms. But basically, some generalizations could be made that if you don't thermoregulate, the likelihood is that since you don't have behavioural plasticity to help you cope with the environmental change, you're more likely to have capacity for physiological plasticity. It also means that you may be locally adapted because different populations of the same species have experienced different seasonal ranges, different thermal variation, and they have the potential to have locally adapted to these conditions. So therefore, thermal optimum for these species may differ between populations. What it means for thermoregulation is that originally it was pitched as the saviour. In terms of climate change, it's okay, you'll just behaviourally thermoregulate to maintain that thermal optimum that you prefer. The challenge is whether or not there's still going to be those thermal microclimates that you need to shift your internal temperature. So it is potentially a risky strategy. And that's because if you've been able to get your optimum temperature with your environment, there's the potential that you then haven't locally adapted. And so different populations of the same species possess the same thermal optimum, and as certain populations can't maintain that thermal optimum, they then lack the physiological plasticity to cope with the changing environment. And this is called the Bogut effect, and it is being found to be true in some lizard species. So the approach that we took is one from the terrestrial literature, and we need 3 pieces of information. We need to understand what the preferred body temperature is of the species, so what's the thermal range that they would like their body temperature to be if they had all the choices in the world. The next we need to know is what body temperatures they actually experience in normal life, in normal reality, going about their business, finding a partner, foraging, getting away from predators. And then finally, we need to know what's the full range of environmental temperatures that they have available. And so this is considered to be the null model for a thermoconformer. So if your body temperature is the same as the environment pretty much all the time, you don't have any potential, or you're not utilizing thermoregulation. If you can shift your body temperature away from what's being experienced in the full suite of environmental conditions, you're a thermoregulator. And so to start this research project off, we've been working with a rabbitfish, Siganus doliatus. We chose it because it's a tropical reef fish. It has a depth of somewhere between 2 and 15 meters. It grows to a decent body size. So as I get onto the methods, we needed a relatively decent body size fish to be able to use the tagging technique. We also know that it has a definable home range and it's territorial, so we have a chance to define the individual that's tagged again. We've been doing this research since about December 2015 at Lizard Island. And just to give you a general thermal context for Lizard Island, if you use the AIMS loggers and just give an average of months over time, we generally see that the seasonal range is somewhere between 23 degrees Celsius and 30 degrees Celsius each year. So first, to test the preferential temperature. How we did this is with a shuttle box system, a 2 figure-8 system, where one side is a cold side, one side is a hot side, and these two sides are maintained at a 2-degree differential. The fish is placed into the chamber the night before, allowed to settle in, and then we run it in a dynamic mode for a whole day. So the fish can shuttle between the two sides, and a camera mounted above it knows which side it's on. It's tracking the fish. If it's on the cool side, both of the sides start to cool down, so they still maintain the same 2-degree differential. The hot side's always the hot side, the cool side's always the cool side per fish. And then if they then switch and swim across to the warm side, the whole system warms up. And what happens is you leave them for, say, 8 hours, they shuttle between the sides, the system goes up and down, and eventually you end up with a situation where they either just swim between the two sides or they tend to sit in the little corridor in the middle. And that's your thermal preference. So for all the individuals that we tested, we found that Siganus doliatus has a thermal preference range of somewhere between 28.8 degrees Celsius and 30.3. We tested them both from a winter minimum and a summer maximum, and they pushed the system either up or down to reach the exact same range of temperatures. To get the information on what heterogeneity we have in the environment, we've been using some Hobo loggers, and this we use to create the distribution of the environment and test whether the body temperature is shifting from the thermoconforming expectation. They're logging every 10 minutes, and we make a big effort to stick them in as many different locations as possible, whether it's out on the reef, in a hole, under a soft coral, to try and really understand what the thermal heterogeneity is out there that these fish could have access to. And then to work out body temperature, we've been using archival tags, which means that you collect a fish from the reef, you surgically implant the tag into their gut cavity, stitch them up, and then put them back out on the reef. We tend to release them within 48 hours of capture so they don't lose their territory, they can get back out there with their partner. We tend to collect both individuals in the pair, hoping that they'll then stick together and have better survival capacity. We've been using both temperature heart rate tags and temperature depth tags from the Star Oddie company. Then what you do, you leave them out on the reef, you come back a few weeks, a few months later, and you're looking for this. You're looking for a couple little sutures that haven't dissolved with a little bit of algal growth, then that's your individual with a tag, and we recapture them. So in terms of the results, so what we've got here is a really good coverage over the 2 years that we've been doing tagging, from what we expected to be the minimum up to quite warm actually, because we are including both the thermal bleaching events. This is a relative density of the measures for either a combination of 12 individuals in terms of the fish tags versus the environment. And I've just put some dotted lines in to help you out with what we discovered was the sort of thermal preference range across individuals. What we see is that individuals, when they're above their sort of thermal optimum range, are shifting to experience these environmental conditions less than would be expected as a thermoconformer. We see that on this side of the curve, when they're slightly below their temperature optimum range, they're shifting to experience these closer to optimal conditions more than would be expected. And you see that with the increased observations closer to that thermal range. Now this is a bit weird. Of course, there's like a peak all the way out here in the middle of 24 degrees Celsius. So I'll just show you another slide to hopefully elucidate that. I've now split the data between daytime and nighttime, and we can see that a lot of that large peak at 24 degrees Celsius is related to the holes that they're sleeping in. I also think, but I'm not going to go into detail today, one of the reasons why we see these sorts of peaks here is that when these fish are that far away from their thermal optimum, there's potentially a benefit to just narrowing your internal temperature so that if you're not experiencing close to your optimum, you might as well just experience as minimal thermal range as possible. But we can see that the same trends are still there, that they're experiencing less of the environment either side of the thermal optimum. And just to break it down in a little more detail, we were pretty excited that we got to see what was happening with the body temperatures when the water at Lizard Island got a lot warmer than it would be a normal summer season. So now I'm just presenting to you the values that we have when the water is above that temperature preference, the temperature set point range. And the environment and the fish are in the same colors again. What we can see is that the fish actively avoid experiencing body temperatures over 32.3 degrees Celsius, and actually probably really try not to get their body temperature much more than a degree or a degree and a half above the thermal optimum range that we got. So that's really interesting, and we are pretty excited by this, but it's possible, but how exactly are they doing it? One of the first things you think of is depth. They're just going to shift off the reef flat, go a little bit deeper, experience cooler conditions, and that'll solve the problem. Well, we do know that we get thermoheterogeneity depending upon where the logger is, whether it's in the shallows or in the deep. So we explored 2 tags that we had over that summer period that also had a temperature depth tag in them. And so the first individual on the left is from the bleaching in 2016. The one on the right is from the bleaching in 2017. We have the depth measures for the tag versus environmental temperature on the top. And what we see is that depth definitely isn't the be-all, end-all refuge for them. And this individual here, he's actually, or she, is using depth less as temperature increases. Whereas the second individual we have is using depth equally regardless of what the environmental temperature is. And maybe this isn't so surprising because these fish are territorial, they don't have unlimited access to the entire part of the reef, they can't just scoot down into a deeper habitat if there's another pair of doliatus living there, so they do have to make the most of the environment within their territory. So the next thing we looked at was environmental heterogeneity. So now I'm just showing you a couple of the paired loggers that we put out. So we have one logger that's on the outside and then we stick one other logger on the inside. They're only within like half a meter of each other. And I just wanted to showcase sort of 2 examples where the grey one here shows that that over time, there is quite a large difference between the logger that's inside and the logger that's outside. Sometimes the logger that's inside is over 1.5 degrees warmer than than the one that's on the outside. So even over very small scales on the reef, there actually is a lot of environmental heterogeneity. The green paired loggers show a completely different result where it tended to be that the outside logger was always warmer than the inside logger. So there's definitely access to environmental heterogeneity, and we believe that this is how these species may be doing it. So just to summarise, there is some capacity for behavioural thermoregulation in coral reef fish, and that it does allow them to maintain a closer to optimal temperature. But we do know that you can't have whatever body temperature you want. Compared to the lizard literature where you've got the option of basking or going in a burrow, there isn't unlimited environmental heterogeneity, but so that when it's winter time, you can't possibly be at your thermal optimum. So maybe this is a good sign. So for future research, we're trying to understand how this links to physiological plasticity. So perhaps that's a positive because you won't end up with this trade-off between adaptation and plastic potential. But we do know that they are able to utilise behaviour to avoid stressful warm conditions, which is pretty exciting. And we know that we believe that they're doing this related to the microclimates that are available in the reef matrix. But the problem with that is that you actually have to have reef for that to be possible. If the environment becomes just a flat space, there won't be the opportunity for behavioural thermoregulation. So the next step with this research is to try and understand how widespread behavioural thermoregulation is. So we have another model species that we're looking into, Tinaquita striatus, and we're also moving forward with another rabbitfish species to try and understand how an environment that has a lot of reef and potential for heterogeneity versus another location that doesn't have those opportunities will alter how the species behaviourally thermoregulates. With that, I'd just like to say thank you and really point out that this research is very much a joint collaboration with Rebecca Fox, and that this was really the ideas of both of us moving forward and hoping to ask some questions. It was largely funded by the Lizard Island Reef Research Foundation. We got one of the postdoctoral fellowships to share, and SeaWorld. And obviously there's other collaborators and volunteers that make it possible. So thank you.